Welcome to the NeuroTools Webinar series. This is the first inaugural webinar in this series, and we are going to try to do these every Friday at 11 o'clock in Pacific time. It's 2 o'clock Eastern, as you already probably know. And in this particular webinar, we're going to talk about RRIDs, which are these unique tags for key biological resources. And the reason that they apply to NeuroTools will hopefully be very obvious to you by the end of this particular talk. So what I'd like to do is introduce my friend and colleague, Dr. Maryann Martone from UCSD. She's a professor, currently Emeritus, but she is still quite active and participates in all of these projects, so I'm not sure what retirement looks like anymore. But without further ado, I would love to turn this over to Dr. Martone.
Thank you, Anita. I hope everybody can hear me and that I will be notified if you can't. I'm just going to give a brief introduction to the RRID project. I'm going to go just a little bit of background, the past, the present, and then Anita is going to talk about the future. So I'm just going to set the context, and then Anita is going to go through some very practical information about how RRIDs are used by authors, publishers, tool providers, and funders. At any time, if you have a question, please, again, put it into the chat, and we will address them probably at the end. But if it's a pressing question for clarity, I'll try to do it while I'm speaking. So before I start, I want to say that first, the NeuroTools seminar series is actually hosted by NIF, the Neuroscience Information Framework. This is a project that's been going on since about 2008 and even a little bit before, and it is an initiative of the NIH Blueprint. And essentially, NIF was established because researchers there was a lot of investment by the government in digital assets. People were creating databases, software tools, all kinds of different projects were underway for creating research resources like animals and antibodies. A lot of this information was contained in databases. And NIF was really charged with essentially surveying and cataloging this sort of new emerging types of products that were now available online. These are things that are not well searched by Google, and as we'll talk about, are not well referenced in the literature. But through the NIF project, we were able to create these very comprehensive data registries that was able to look across the entire landscape of research resources, which again, we define as reagents, animals, databases, software tools, training opportunities, all kinds of things. So this project is both hosted by NIF, but it is also enabled by NIF. It was the NIF project that first started to identify problems in the literature with identifying research resources, and also proposed a solution for how this could be solved. I also want to acknowledge DKNet. DKNet is funded by NIDDK, National Institute of Digestive Diseases, Diabetes, and Kidney Diseases. And it was through DKNet that we were able to help launch some of the infrastructure that currently supports the RRID project. So I wanted to acknowledge these, and also if I use these terms, wanted people to know what I was talking about. So essentially, what are we talking about with RRIDs? Research resources, which again, we're defining as anything that one uses to produce the results of a study, are essentially what we call the ingredients of science. And we all use these, every paper reports on them, yet there's a problem. And this problem is that in the age of global search, Google and everything else, we should be able to answer two questions very easily wherever science is done or communicated. The first is, what resource? What is the exact resource that we use to produce the results of a study? And secondly, what other studies have used these resources? So we should both be able to identify, and we should be able to track. Well, why do we want to do this? Identifying the reagents we use and the resources we use is essential for rigor and reproducibility. We know that these key resources like antibodies and animals and even software tools are key sources of variabilities inside of individual studies. And so one would want to know, the first thing is, did you use the same antibody? Did you use the same animal? But you also want to be able to track impact. These are often funded by government initiatives, and one would like to be able to, if you're either a resource provider or a funder, to be able to say, hey, how many people are using my resource? You want to be able to give credit to people who do produce these things. Right now, you get credit for writing your papers, but as many of you who may follow this know, there's a movement afoot to give broader credit for the sum total of things that we do that can actually have impact. You want to be able to aggregate data. Essentially, the biomedical literature is one gigantic meta laboratory, right? The places where people take these animals or these antibodies and they put them to use in all these different studies is reported through the literature, and you'd like to be able to find all the papers that use a particular mouse or all of the papers that use a particular antibody. And finally, you want to be able to monitor the health of the scientific food chain, as we call it. If you've been following some of the stories about reproducibility, you know that sometimes antibodies don't work as, well, even if you're a lab practitioner, you know antibodies don't work as advertised, or that cell lines get contaminated. And currently it's been documented that these things still get reported in the literature because there's no good way to notify people or notify readers of a particular article that a problem has been reported. So why can't we do that now? Well, again, RRIDs are saying we can do that now, but essentially scientists don't report enough identifying information to determine what exact resource was used in a paper. I think we've all experienced this, and our current systems for actually reporting this are non-standardized. A lot of people are putting things like locations of vendors because that's how we were all taught to do it, which is not very useful these days. But when you don't have a standard way for referencing research resources in a paper, it means you have a hodgepodge, and it's very difficult to be able to pull out any information unless you actually have access to sophisticated natural language processing and others. We contrast that to our current citation system where, imperfect as it is, if somebody cites a paper, we're able to pull back that this paper was cited across all these different papers. And so having a standardized system is very helpful. And these are just some examples. Anita's going to tell you more, but you often get these statements that say, hey, I purchased these mice from Jackson Laboratories, no stock number, nothing. You go there, you can't find it, or you find 50 mice that all seem to satisfy the search string. So essentially, we know this, that people don't do a particularly good job of identifying information in the paper. Here's another example, again, FICC anti-human CD19. A lot of these antibody companies go out of business, they rechange their stock, all kinds of things. And so the identifiability problem has been well known. So how common is this? Nicole Vasilevsky and colleagues back in 2013 published a paper where they actually looked across a lot of different resource types and looked at how many you had enough information to reliably trace that back to a single offering in a catalog or a single offering in a stock center. And you can see that it's not very good. Some things are better than others, but generally things hover around 50% or below. So can this be fixed? Of course it can, or we wouldn't be here today. How? In the age of global search, you have a unique identifier, something that a machine is very easy to search on, that uniquely identifies each one of these research resources. We call this identifier an RRID, and basically when authors refer to these in their methods section of their paper, they put in this syntax RRID followed by the unique identifier, which is given by a database authority that's responsible for it. And that makes it very easy for us then to track all papers, identify this with search. And again, Anita's going to go through that in just a minute. But I do want to also here give some, just a very brief overview of the history of RRIDs, because we get a lot of questions as to why we did things the way we did. And I think it's really worth mentioning that the original problems with resource identification went way back to 2009 when NIF was just starting and trying to figure out ways of pulling research resources from the literature. We realized we really couldn't do that because there wasn't information in there. Meetings with LAMD, which is Linking Animal Models to Human Disease Initiative, which was going on around 2010 and 11. When you met with the curators from major databases like the model organisms, you realized that this was an endemic problem. And also the discussion with NIH program officers of saying, we're funding these things, but we don't know how to find out who's using them because they're not covered by a citation system. So essentially this project was in the works for almost 3 or 4 years before we were able to work with the journals and others to propose a solution that was mutually agreeable and actually get that into practice. And the exciting thing is that even though this started in neuroscience back in 2014 with Journal Neuroscience and Brain Behavior, Journal Comparative Neurology, as Anita will show you, it has continued to grow and grow beyond neuroscience. So the Endocrine Society, for example, is now using the RRIDs. And so we think that that means that we've, again, identified a problem that is endemic to all of biomedical science, but also provided a solution that was easily implementable. And that's what Anita is going to talk about in the rest of the presentation. So thank you.
Okay, thank you so much, Maryann, for your comments. And so really what we want to do is we want to ask, what are these RRIDs? And this is a research resource identifier. That's what it stands for. And the way that we think of it, and the best way to really think of it, is it's this barcode for scientific reagents and other kinds of tools. And so what you find here is that if you look at some of the vendors, the companies that actually have these kind of research resources, so here's BioLegend, there's a nice RRID for this particular antibody. Now this is on their product page. MMRC is a nonprofit run from various universities. This is a strain data sheet, again, talking about how do you cite this. There's the citation ID. Here is the RRID right there. And so the scientists will actually take and purchase these reagents or obtain these reagents in some other way. They'll do their experiments, and when they're ready to report on those experiments, then the publishers often will ask these researchers, hey, can you please figure out which RRIDs you're actually using in your paper and put those in. And then here's just an example of another paper with an antibody that's actually cited by the research resource identifier. And these papers can then actually be used because these are very standardized. They're actually clickable, linkable, linked objects in that paper, and these can even be brought out into apps and other things which can bring information from the databases back into essentially this paper where that person is reading. So, but we often get this question, okay, so there are different ways to kind of name that particular object, right? So any cell line, any organism, any antibody will have a name. It'll have a stock number or catalog number. Why do we really need this RRID? So let's look at some examples. So here's an example of a stock number, 25. How do we know that's a mouse? MAB 377, you might know if you're using antibodies that this is actually an antibody, but otherwise it's a little bit iffy, right? But if we look at the RRID syntax, right, these actually are a little bit more consistent. So first, RRID basically means, hey, I am using this thing, IMSR, CRL, these are prefixes to a particular mouse, right? So when you know how to read these, they actually come up and become a little bit more meaningful. The names are the most meaningful, but they're very, very difficult for search. So the way that we can look at this is the stock numbers are locally unique. The RRIDs make that same thing globally unique. There are also 4 different people, right? So the name can be read very accurately by curators or researchers who are really, really well trained in how to read, especially the genomic nomenclature. The stock numbers are meant for customers, and for RRIDs, they take a lot of these stock numbers, they add on to them, and essentially make them globally unique, which makes it very good for authors and for search engines. So they try to answer different questions. So the citation, how do I cite this thing that is used versus what is this? These are different questions that these answer. But of course, we need to understand that all of these are part of the same whole. So really quickly, let's just look at this this actual anatomy of an RRID. So here is a particular zebrafish that comes from Zerk. Its number, its stock number, that's the local identifier, so it's 1515. And so this is what it looks like. Now the anatomy of the full syntax, this is a particular antibody from a company called Millipore with a local identifier here. We suggest that the lot number is used. And of course, the global identifier from the authoritative database is at the end. So what should authors do with RRIDs? Well, as I suggested before, there's a workflow for authors. So the journal actually will direct the author to a website at SciCrunch. This is a shortcut. So rrid.site is a shortcut to get to SciCrunch. The author then will search in the search box. And in this case, they're searching for 108L. Catalog numbers, stock numbers are very easy to search for. And so 108L actually corresponds, there are two different results. So that's actually fairly typical for catalog numbers. But then there is a little Cite This button that opens up into a box that can be copied and then pasted into the manuscript. And as the manuscript is published, it then actually is searchable by Google Scholar, by PubMed Central, or anywhere else you want to search. And so now that identifier can be searched by very, very simple means. The RRID portal itself doesn't just bring back ambostoma, it actually brings back a lot of antibodies, millions of antibodies, about 500,000 organisms from various stock centers. I'll show you some of that. Lots and lots of cell line. We're nearing 100,000 cell lines and a lot of software projects as well. It turns out that when asked, some of the eLife authors that had published papers with RRIDs were able to say things like, well, adding the identifier turned out to be very straightforward. They think that it's helpful to enhance transparency of not only our paper, but also countless others. So people see this as a pretty reasonable way to actually put a lot of rigor and transparency into their papers. And this one is very interesting. This is Dr. Dutta from University of Virginia, who basically said, besides helping other labs, our own lab is also going to benefit. So that's a great thing. So it seems to be beneficial for the author themselves. And then when does this really become critical? So in some cases, there are not a lot of cases, but there are certainly some cases where there are problems that are already known, and some of those problems aren't necessarily reflected. So this is an example of a search for 1-5C-4. This is a cell line. And this cell line is actually known to be contaminated. Now, that's known to be contaminated at the source. It's not a contamination in a particular lab. This contamination happened at the source. So HeLa cells actually overgrew this particular cell line. So if you're about to publish a paper with this cell line and you don't know about this contamination because you bought this cell line from a vendor that does not reflect that information that this is known to be contaminated, then this is a great last line of defense just to ask the authors, hey, wait a minute, did you know that this is contaminated? Now, it may very well be just fine that this is a contaminated cell line, but then in the discussion section, one should address the fact that, this is in fact a HeLa cell. It's not what we thought it was originally, but our results say X, Y, and Z, right? So this list of cell lines is actually derived from a list put together by ICLAC, which is the International Cell Line Authentication Committee. And this has grown to well over 700 cell lines, and it continues to change. So this is something that needs to be checked periodically. But at last, right before the paper's published would be a great last check. So this is definitely that place where it becomes critical. So journals, why should they care about RRIDs? Well, it turns out we make it really fairly easy to go across all the kinds of things that are considered key biological reagents, including obviously antibodies, including cell lines, including all the different kinds of animals that that people are commonly using. Now, we don't have absolutely everything, but we certainly have a whole lot of it, and we have a goal to be cover as much as possible. All of that is put into one place so that the researchers, the authors, can actually search only in one place as opposed to searching in 20 or 30 different places. Then the other thing is that there is a master copy. So the catalog number doesn't always stick around at the manufacturer. The catalog number doesn't always stick around in the stock center, but we keep a copy of everything that's out there. And so if there is in this case, there's a fish, this ZL1515 is a fish that's no longer available from Zerk. But that's okay. We can still have that in our labs and we can still publish with it. And when we go to our RRID portal, actually both the master copy of this data and the new data are both reflected in the final list. So data persists even if the fish or the mouse or whatever stock of whatever cell are no longer available for purchase, because often these things actually stay around in individual labs, or the paper stays around in an individual lab for many years, and the stock may not. Okay, so that just says that. Okay, the other reason for journals to really like this is because when we've looked before the pilot project, and the pilot project was, hey journals, can you please put in some RRIDs? And the issue of the particular journal just before the RRID project was considered to be pre-pilot. So we took a look at all of those papers and to determine how many antibodies could be found and could be understood. And the average was around 50%, which is exactly what Vasilevsky saw in 2013. And then the first issue, where there were papers with RRIDs, and looking at those papers with RRIDs, we could determine that, hey, we actually found a whole lot more antibodies. So this really means that actually, when you ask for RRIDs, you end up getting much better data, much better information from the authors in terms of at least identifying what they're using, if they're being asked to do this. So we win. Right? We win. We make resources more identifiable if we add RRIDs. Here are some nice more examples. These are two mice, both from Jackson Labs, and you can tell that this is coming from IMSR, which is the International Mouse Resource Strain Resource, from the Jackson Laboratory. There's its stock number, and actually if you click that, you can get back to a place where you can actually find the mouse. Whereas if we were to label it like this, which is what we saw Maryann talk about before, here would be another way of trying to cite the same mouse, but it's not as good. We like the stock number, right? So how can RRIDs really help resource creators? So we're here on the NeuroTools website. We have a lot of people who are going to be talking about NeuroTools and people who are going to be trying to use NeuroTools. So resource creators can be helped by RRIDs. Now, without any fancy tools, as Maryann had alluded to, without any other kind of natural language processing or anything else, every one of us can go in with an identifier, RRID, in this case, this is an antibody that's fairly commonly used, but they can go into Google Scholar and they can do this exact same search. And they can monitor, they can set up a monitoring system for who is using my tool. So that is a very easy thing to do if we have the cooperation of the authors and the journals. But we can do better than this, right? So there's an entire system of these things called identifiers. And these are all handled by Citation. So we know that actually papers have DOIs, they have PubMed IDs, they have PubMed Central IDs, and we're kind of familiar with these now. Resources have RRIDs. So these are the objects that we use in methods. But authors have ORCIDs. They also have a slew of other identifiers, but we have ORCIDs, and we understand these. Data sets get DOIs, funding information get grant numbers. Now, the goal of the RRID project is not just to create RRIDs for all of these things, not just to get these used, but we can actually start to link papers with resources, papers with authors, resources with authors, resources with grants, and on down the line. So really the goal is to start to create this ecosystem, which is much more machine readable than our current system. So let me show you some of the things that fall out of actually using identifiers. So this is the same eLife paper with these 2 mice. And one of the things that we've been able to do, because we have some graduate students that can write a little bit of Python, what we can easily do is create actually this little Cybot, and it's real, it doesn't really look like that, but it's a few lines of Python. And with those few lines of Python code, what we can do is we can actually go in, when somebody looks at the paper, we can actually run a script that quickly reads the paper with us and for us, and identifies these things that are RRIDs. And in this case, it's actually taking this identifier and it's asking the database, hey, what do you know about this thing? And here is what the database says. And the database says, okay, it's stock of this ERC mouse. Its affected gene is ERC1. It is currently available as cells. This is its catalog number. And then if you click on the database, you can actually get back to the strain data sheet, and you can buy the mouse if you wish to. Now, this might seem trivial, but it actually is not. And you can actually see these examples looking at this. So if you just go to PubMed Central, just type in 4107038, and you can see some of these if you have the hypothesis plugin. But if we look at any of these kind of structured annotations, what they're really doing is they're saying, hey, there's a paper, here's a paper, this one's in GigaScience, and this paper is now associated with the tool. So we're starting to make those RRID paper associations. And then what we can do is we can then import that data. Now this is within our system. So there's a particular tool, Abyss, being described in this paper. And now if we look at Abyss, the web page for Abyss, and we looked at this area which is referenced by, now we see that this GigaScience 2017 paper, it's this one, actually has been associated with this tool. So this data actually fills, the RRID data fills the resolver and is made openly available for scientists, for publishers, for anyone who wants this particular data. But how much data is there, right? And what is it, how does it look? So if we look at this, RRIDs as of this morning have been noted in 4,373 papers. We're really hoping to get to 5,000 in the coming weeks. These are coming from 349 different journals. So that's actually, it's starting to get up there. Now, if we look across and just look at the number of papers that a particular tool was being used in, even though most of the RRIDs are for antibodies, the most common tools being used are actually in the software category. So that's these guys. Quite a few now cell lines, but in the top 25 list here, there's only, I think, one cell line. Almost all of these things at the very top are things like ImageJ, Prism, MATLAB, SPSS, so these statistical tools which are starting to be used in hundreds of papers. Which is actually quite interesting if we think about it. So what does one of these pages look like? So if I am the creator of ImageJ, now what I can see is, I can search for ImageJ, I can click on it, and here's the page that comes up. So this is a great neuro tool. Obviously, we see that it's used in many hundreds of papers. This is our most popular tool. And the first thing you'll see is obviously there's usually an icon if we can find one, the name, the RRID, of course, URL, a small description, and then there's various other information on the first page. There's some relationships to other resources. And then if there's a paper about ImageJ, which there are now several, although there were many, many years where there was no paper about this tool, that would be listed here. And then if papers, yes, Maryann?
We have a question about DOIs versus RRIDs, so make sure you cover that.
Oh,
okay.
DOIs versus RRIDs. That's a very good question. It's one that does come up a lot. Let me finish the referenced by section, and then we will. I'll move on to that question. Okay, so if there are papers that actually use ImageJ, they would be, if you click on the referenced by, those papers would be listed here. Now, what we'd love for all tool creators to do, and so if I were the creator of ImageJ, I would click on this button to log in, then what I would do is I would claim ownership. So click on that, and then you have to write a little, 1 or 2 sentences to tell us who you are and why you think you own this particular resource. Then that would be great. You could also subscribe to new mentions so that when you come back to this site, there will be a little ping for you that says, hey, we found 3 new papers, or whatever number that happens to be. Okay, the second piece is an association with ORCID. So if you actually go in and you're logged in, you can click on the My Account and on the Information tab, and then here in the green box, there will be a little box here, actually, underneath your name that says Associate ORCID. And so for my particular user account, I've already associated my name with my ORCID ID. So I validated through ORCID. And now the question is, what does that do? Well, we are starting to work with ORCID to actually, expose some of these things that may not be traditional academic or scholarly works. Some of these things, such as I created ImageJ. If I created ImageJ, that would be great because I would be very famous indeed. However, I have actually, I'm one of the creators, probably the critical creator for the antibody registry database. So now my ORCID record is able to reflect that. So here is my ORCID record, and it includes this particular work, which is a type of other work. This is not a paper. It is not a conference proceeding. It is a work that has an RRID and a URL. And in the near future, we will actually be pushing anything that is associated, any tool, neuro tool that is associated with a particular ORCID back into ORCID, of course, with the permission of the author and creator. So please do that if you have a tool. But actually, we're doing even more. So we're working very closely with both Hypothesis and with Crossref on their new event data database. And what this means is that if there is something about a paper that happens after a particular paper is already published, and one of the things that happens is our robot actually finds the RRIDs in that paper, that is now able to be noted by our robot, and we export that on a relatively periodic basis through a mechanism that Hypothesis has worked out. So we export that data to Crossref. So now Crossref knows that a particular paper, which has a DOI, is associated with a particular mouse. And in this case, this is just an excerpt of the back end of this particular data, which shows that, hey, this paper, which is described in this JSON blob, has, uses this particular RRID. This is a mouse from Jackson Laboratory. Its ID is 6664. So this is, I happen to know that is the C57 Black 6 wild type mouse. Anyone can actually hit this. So this is the this is a method of going to that particular event data database at Crossref. So we don't control this data, but we make it available for developers to go ahead and hit. But this actually also, and at this point, I would love to be able to really address this notion of, well, if we're going to have a DOI, that DOI is for a digital object. RRIDs are mainly not for digital objects. They're for mice, they're for antibodies, they're for cell lines. And mice already have very nice identifiers. They're Jackson Labs identifiers for the stalks. They are MGI identifiers for the full genotypes. It would not be appropriate to put a DOI on a mouse. The same as it would not be appropriate to put a DOI on me. I am a human. I am governed by ORCID. Software tools are a little more finicky, right? Some of them do have DOIs. They all have, RRIDs. So maybe, there's a little bit of overlap there. But a DOI does not talk about a project. A DOI really talks about the version of software that might be used. So in the case that there is a MATLAB script that is being published alongside of a paper, then that MATLAB script should get a DOI because that is the exact version that was used in this paper. But if that is not a MATLAB script, or it's not a Jupyter notebook, but that particular software tool is something like ImageJ, which has multiple authors over 25, 30 years now, with multiple grants that are associated with that, with the institution that's associated with that. Well, that's more of a software project, which would then be covered under an RRID. So that's where the differences are. And actually within the full syntax of the RRID, there is the place to put in that deeply identifying information, the lot number, the version of the software, or the DOI of the software, along with the RRID at the end. So that is how we're approaching this intellectually. Okay. So the numbers of papers is really growing rapidly, especially coming in 2017. But this really reflects, so from 2014 to 2017 is when this project has been ongoing. 2017, these are relatively current numbers. But what you can kind of see is that there's roughly a doubling every year. So if we continue with this doubling effort, then we know that within 5 to 10 years, we should actually be able to have roughly all the journals that are indexed by PubMed, and hopefully a good chunk of those papers. If we look at the current data and we look at it by publisher, we can see that several publishers are really kind of overrepresented, and lots of other publishers are sort of underrepresented, and this kind of trails on. But really, if we look at who, which publishers are actually have the most papers, then we can get a really good sense of, who is actually enforcing the standard on a regular basis with each of their papers. And I will say that, both the several society journals, eLife definitely, even though it's come on relatively late, is enforcing very strongly. And of course, Wiley, there are several really great Wiley journals that are pushing this bar, and they really started this. But Cell Press has brought on all of their journals recently. Again, this is very recent, but this bar actually reflects the fact that they are doing this very consistently. And Oxford University Press also does that. Several journals that are doing this regularly. So the data doesn't seem to be very big, right? It's a few thousand papers. Why didn't we just do that by hand? Why do we need robots? Well, looking forward in the next couple of years, if we do not have robots, there is almost no way that we could handle the volume that comes out monthly in PubMed. So the volume, if you're not aware of this, is about 1 to 2 million papers a year, or 40,000 a month, right? that volume is not a volume that any human can read. Even if you have multiple humans, they can't read this. So we really do need to work on some kind of an automated means. Bots are great because they actually reduce a lot of the curation time, and time is always money. So they are also much less error prone. The syntax of what comes out is always correct. And they do basically allow the lag between the HTML version release of the paper and the release of the RRID annotation data to be about 1 week. So we can actually keep up with the literature as it's going now, when everything is correct, down to 1 week. So in publication timescale, which is somewhere between glacial and I believe epochs, that 1 week is really, it's the blink of an eye. So we're very excited about this number. So we are also starting to see some of these kinds of bots being utilized at the publishers. Okay, so what is the future? So really quickly, what I wanted to just briefly mention is that we can actually improve the tracking and the impact of some of these resources. The more authors that do a better job actually identifying their own resources that they're using, then we can really move all of those papers from that 50% identifiability to 90% identifiability, and that is absolutely great. Furthermore, with the 90% identifiability, it's not just identifiability, it's machine identifiability, which is better than human because it's more consistent and we can actually start to use that data. We can start to build the system that actually provides credit to tool makers, not just writers of papers, but actually writers, makers of tools. Today we can do this with software resources. In the next few months, we'll be working on the same kind of ability to link to ORCID from antibody providers so if a person makes a bunch of antibodies, those antibodies are used in hundreds of papers that ought to get credit for that intellectual effort of making a great antibody or a great mouse or what have you. But we can even do more. Because if we look at something called Resource Watch, which we're calling Resource Watch, we can start to track problems with some of these key biological resources. And for example, we have the contaminated cell lines. ICLAC already has a great list. Could we put that information into PubMed or into PubMed Central? And the answer is, of course we can, as long as we can identify most of those resources in that paper. So we can provide that information to authors at the time of publication, as you saw. We do have some evidence that this information is read and used. And of course, we have a means to actually disseminate any problems that are reported after the author has published that paper to readers through this hypothesis mechanism and also through Crossref, which would have the same data. So, we thank you for being here very much. We really appreciate your being here, but this is really a call to action. If you have a tool, a NeuroTool, we would love to know about it. We would love to see if we actually already have it listed on the website. We want you to control your tool, so we want you to click on that. I'm an owner, and we want you to associate your ORCID, because most people who write tools, at least for software tools, really do not publish papers until well after that tool has already been used. And sometimes it's been used. So I think the first paper on ImageJ wasn't published. It was published more than 10 years after the first papers were actually already using ImageJ. So there are many, many tools that don't have references. So this is a mechanism for you to get credit. RRIDs should be in your methods section if you are an author. Many people review, I review, and when I review, I try to review from methods. If you review for methods, please ask those authors to add RRIDs. And of course, if you happen to have a journal, we would love to get you on board. So any comments? Please send them to this email address, then you will get myself, and I will forward those comments as needed. So again, thank you all. I believe that we have a few minutes. We have about 10 minutes for questions.
Hey, Anita, that's
Yeah.
correct. We have a little bit of time for questions. And first of all, thank you to Dr. Bandrowski and Dr. Martone for an excellent presentation. And thank you to everyone who is joining us today. How we'd like to handle the Q&A section, if you could go ahead and type your question initially into the chat box, that would be most efficient. And then from there, we can work one by one and unmute you if you feel like you need to elaborate. And as we work through the question list, that'll help us avoid too much chatter in the participant section at one time. So if there are any questions, once again, now would be a great time to type them into the chat box. Dr. Pollock, I believe, had a question earlier in the chat, but my understanding is that it was answered. If it was not, Dr. Pollock, please feel free to elaborate now.
So again, I think that this question really was, the question of whether or not DOIs were the appropriate mechanism as opposed to RRIDs, and what are the real differences. I think the best way to answer this question is an RRID would be a project, or it would be a mouse, it would be something physical. It's not necessarily a simple digital object that's relatively immutable, which is what the goal of the DOI is.
Right.
Do you have anything to add to that, Maryann?
No, we get this question a lot, and I don't know how many of the audience are in the sort of publishing world. So one thing I tried to type into the chat is that if you're in this world of identifier systems like DOIs, you may care a whole lot. If you aren't, you may not care at all. So, but essentially, the recognition that the way that the digital world works is on these unique and persistent identifiers. And this persistence, as Anita talked about, is that there's an organization who stands behind them being persistent. There's nothing magical about them. But what makes them powerful is their uniqueness. So whereas anyone can just go and register and get a DOI, if every DOI points to something different, then they're not really helpful for aggregation. And that RRIDs were really, again, directed towards the two questions I brought up in the beginning. What did you use? Very compact way of identifying that, and who else is using this? So you want to be able to say other people are using it. So the flip side, as Anita said, to the DOI is the lot number and batch number and all of the specific information that can go into an antibody. And people said, well, how come you don't identify that? And first of all, again, as Anita said, all of that information should go into the paper. It's absolutely essential that it go into the paper. But if you start to overload the identifier systems, they become very complicated and unwieldy. And they don't let you do that first pass that said, okay, I just want all the papers that use this mouse. I know every mouse is different from every other mouse. I know every time a strain crosses over to a laboratory or to another stock center, it migrates. We've had lumping and splitting since the beginning of time in biology. But the important thing is you want to be able to ask that global question, the global question. So it's the same with our software and everything else. Whereas an article, a DOI points to the single article. You'd be in different forms, right? But it's the same article. We wanted something that pointed to all of MATLAB, not just that particular script. So that's wwhy it was identified.
And Anita and Maryann, I just want to make sure you guys are seeing in the public chat. Dr. Pollock, has a follow-up question. How would you specifically cite DB gap in a paper?
So good question. DB gap has an RRID. And if you put in a method section, I got my data from DB gap, you would put the RRID in right after you said DB gap. And then if you took a specific data set from it or a specific subset, you would give that subset of data, perhaps a DOI, and cite them both. Yep. So that's how we would do it.
Yeah, I absolutely agree. If there is, if there's a particular database that actually gives persistent unique identifiers, I mean, we know some of these from the genetic world. We know geo-identifiers. These things are great for identifying a specific data set. So if dbGaP itself does have an RRID, which is great, it's associated with the creators, the grants, and what have you. But a particular data set in dbGaP is associated with an identifier from dbGaP. It might be a collection, might be associated with a DOI, which then resolves out to all of the different subsections that the author took. And but for Geo or dbGaP or one of these big well-known databases, they tend to all have unique identifiers for the particular data set that someone is looking for. And that should absolutely go into the methods section. And there's actually a section that is now accepted as part of the references, which is data.
Right.
And that is that's something that's gone in. Does that answer your question?
It's a subtle distinction. And as we've talked about with the identifier people, we live in an analog world, not a digital world. And at the edges, there's some overlap. But the important thing is that regardless of which identifier system you use, that you use an identifier system to cite the use of your research resource. And the rest of it gets worked out on the back end and through practice. But it's a very simple way to say, I would like to see all studies that published with dbGaP. Mm-hmm. So I don't care which particular data set they used. I just want to know how many people are publishing with dbGaP. RRIDs make it very easy to ask that question. Mm-hmm. And that's really the point of them.
Okay, so it looks like we have a question from Dr. Ilam. Mm-hmm. Okay, so what about shared data projects like the Human Connectome project, shared neuroimaging data? Should we have an RRID?
And you already do.
We have trouble tracking papers that use HCP data. Yes, we know. This is why we have this.
Yeah.
Some publications cite our NIH grant number. Others cite the resource in other ways. Some journals tag the citation often in acknowledgments or a footnote. I know. In PubMed, others don't. How do you obtain an RRID? Great question. And actually, I can just show you how to search for this. So,
right. So basically your question is exactly why we developed our RRIDs.
Yes.
Is what the question we get oftentimes from people who have projects or who have databases. Yeah, I'm
really not sure why there are little
That's interesting.
Marks on this.
It's Zoom. It's all where you drew.
Yes.
Yeah.
I think I drew something. Okay.
So they say, why are you giving us another identifier? Why are you doing this? And we're like, no, we're giving you a citation format is what we're giving you to encourage authors to document these things in a consistent way inside of a paper. But here is something that's very specific, right, that we wanted to make sure. We wanted to make sure that the RRID system was used in the materials and method section. That is, this is not any mention. So there are papers which say, oh yes, there is this thing called the Human Connectome Project, and they may discuss you, but we really felt that it was necessary from a scientific point of view and an impact point of view to be able to track the actual usage of these things. So essentially, the NIF project was responsible for cataloging projects like yours. And every time a new project came into existence, we would go ahead and we would put it in our database and we would give it this unique identifier. And that's how essentially we started being the ones who issue the identifiers for these types of things.
So I think this is the right project. It's Human Connectome Consortia. So this is the core project. I think it has some relationships. So let's look. Yes. So it has some relationships to different tools, different projects. It's got a relationship to NITRIC, for example, which will be discussed next week. It is referenced by many, many papers. It's referenced by about 440 papers, but some of those are at a lower confidence than others. But we can look at some of the kind of verified ones versus the ones that are not by just clicking on this this confidence. The general information is here. There should be funding information here, which I don't see. So Jennifer, please log into this take control over your resource and add some funding information. So that will then, connect up this particular project with a funding mechanism that used it. And then we have funding from all of these papers, so then we can start to look across.
But an important thing that you can do, Jennifer, is for projects like the Human Connectome, which have a lot of different pieces to them. So I know that there's individual data releases; those all have their own identifiers now. But it would really help for the repositories to help support this citation standard by giving on your homepage. If you have used data from the Human Connectome, please put Human Connectome Project, RRID : and you should let us know what is your preferred one. And you should let the people who are going to cite you know which is the preferred one.
Yep. That is definitely the
So it would be a very helpful thing for us. Yeah. And perhaps we'll follow up afterwards so that Anita can go through these tools a little bit more slowly.
Right. Yeah. But if you do click on the co-mentions heat map, that's in the analytics, what you find for your Connectome project is, because there are 440 papers that talk about different tools, it turns out that these are actually the tools that are most commonly co-mentioned in papers with your project. Mm-hmm. Okay, so we have another question. It's how will journals determine if all resources used in a particular paper include their associated RRIDs? That's a great question.
It is a great question.
Do you want to take that one, Maryann?
So right now,
coming from the journal editor perspective.
Okay, so there's two things. There's different strategies, and this was done deliberately with the with the agreements of the journals of how they wanted to roll this out. So people ask for RRIDs at different times in the process, during review, before submission. But the big question is, well, what happens if I have resources in there that don't have an RRID? And do I need to create RRIDs for everything? We believe that that first of all, tagging the world is impossible, right? So that the idea that you're going to be able to give these unique identifiers down to every level of granularity is that 100% compliance is probably not going to be possible. So if right now we ask the authors to do the best job that they can. If there is a software tool or a database or an antibody that doesn't have an RRID in it, there is a means to submit it so that you can get one provisioned on the fly, because generally authors are doing this while they're doing revisions of their papers. They're going to the portal. If it's an animal or a stock center, you have to go to the appropriate authority, and we tell you go to the appropriate authority. It may take a little bit longer, but you have to be practical with these things. So if somebody's got to get this revision in and it's going to take them 3 weeks to figure this out, they're going to pretty much identify the things that they can identify. That's the beauty, however, of the little SciBot that Anita pointed to, is that the robot, once it knows that there are RRIDs in this paper, it goes through and says, you know what, I know about this other tool, I know about this other tool, the authors forgot to put it in, so we can sort of supply that as metadata on top of it when it is clear what the authors were talking about. Mm-hmm. Because as I mentioned before, the edges of these things become a little tricky. And so we often get the question, well, I should identify every salt that I use, should I identify every reagent? And we say, no, really this is designed for resources that are known to be sort of key sources of variability. And, I think that the practice of what gets identified and when and how and what the expectations are, those sort of norms develop over time. So we don't claim right now that in our data set, and in fact you saw in the Vasilevsky graph there on identifiability, we don't get to 100% identifiability, but we do far better than the 50% that we had. And I think as these things come forward, it evolves. And in the beginning, we didn't deal with secondary antibodies, for example. And then everyone's like, well, we think you should deal with secondary antibodies. And so people start to add it in, right? The community itself kind of helps us decide which things they think need to be identified or which things they don't. Mm-hmm. So I don't think it's a completely satisfactory answer, but the nice thing is, and this is why we did this in conjunction with the journals, right? We worked with the journals at every phase of this. And you don't want to stand between an author and their paper getting published, right? You can't have weeks and weeks and weeks. And so you have to compromise at some point and say, we do the best we can. We'd like that percentage to improve all the time. But if you forget some right now, it's like, well, we're still better off than we used to be.
Absolutely. And so I pulled up an example. This is a very recent paper.
Mm-hmm.
Let's see, I think it was probably published last week. It's towards the bottom of our list. So it was published on September 13th. So this is quite new in Neuron, and there are some nice antibodies I see. But you can see that the compliance is not 100%. Some of these antibodies were not registered. They could have been, but they weren't. So there are a few here that are missing, but the bulk of them are in with identifiers. And again, these things are linked. This is the little app that comes up on the side. And this tells you, in this case, for example, this is a UN antibody from Millipore. Here are some other papers actually that also use this same antibody. So that kind of helps to track this down. Wow, this is just writing all over the place, isn't it? But in any case,
it's kind of cool, but it's distracting.
It is a little distracting. But the organisms are in here. They're not yet linked. They will be. And then the software tools. So in the cell papers, actually, there's a nice list. And ideally, there's nothing stopping any author from putting in a list of all of the things that they used. Some journals make this kind of a table required, but it's really nice to have this list so we can actually look at what are all the different tools that are being used. Often we register these. And then if you actually have the hypothesis plugin for some of these papers, you'll be able to see there are no public annotations, but there are certainly some private ones. So you can see that there are 39 annotations on this one.
Hypothesis will be the subject of another webinar.
Oh, they totally do.
Yes, because we've mentioned it a lot,
Yes.
and I've tried to put some explanatory information in the notes, but it's another one.
It's another one.
Yeah.
All right.
Okay.
So I think this actually brings us up to 12 o'clock,
Mm-hmm.
which means that we are out of time. But I thank you all very much for sticking around, and I thank you for listening. And go out there and get your RRIDs.
And don't hesitate to contact us.
Always.
Yeah.
Thank you.